Hi, everyone. Happy Friday, and welcome to the Myotonic Dystrophy Foundation's Meet the DM Drug Developer session on April 2nd. Today, we are meeting Expansion Therapeutics. As a reminder, MDF's mission, care, and a cure is to improve the quality of life of people living with myotonic dystrophy and accelerate research focused on finding treatments and a cure. Our work focuses on support and education, research, and advocacy. Please feel comfortable visiting our website at myotonic.org to find toolkits and publications, including clinical care guidelines and recommendations. Our virtual support group and Facebook chat information is available. Our calendar of events and activities is posted, as well as access to our digital academy, which hosts presentations and videos from all of our past events, including today's presentation, as well as anything that's happened at our conferences in the past years. The first Friday of each month, like today, at 12 o'clock Pacific, we are meeting different DM drug developers. So we welcome you to join us next month on May 7th for Ionis and Biogen. And although most of these are happening on the first Friday of each month, please note that we do have one bonus day where we're going to be hearing from Artex Biotech on a Wednesday due to the high demand to hear from as many drug developers as possible. You can sign up for any of these sessions at myotonic.org/meet-dm-drug-developers. Similarly, we have a fantastic series happening the third Friday of each month at 12 o'clock Pacific, and this is our Ask the Expert series. So we have on April 16th, which is just 2 weeks from today, we have an expert talking with us about gastrointestinal impacts of myotonic dystrophy. You can register for this or for sessions on the heart or speech and language and a couple others that are in the process of being confirmed at myotonic.org/ask-expert-series. I hope you all know now we are officially confirmed. We have declared International Myotonic Dystrophy Awareness Day for September 15th. You can join the Global Alliance at myotonic.org/international-DM-day. And we will be posting more about what you can do to get involved leading up to and on September 15th. A reminder, today's session is being recorded and will be posted to our digital academy at myotonic.org/digital-academy. Please feel comfortable reaching out to MDF about this or any other presentations. Our phone number and our email address are listed here. Okay. What you really want to hear about are today's presenters. So Dr. Ronaldo Scarole is the CEO and president of Expansion Therapeutics. He has over 30 years of pharmaceutical experience in the discovery and development of small molecule drugs to treat cancer, infection, immune-mediated, and rare diseases. He's an accomplished drug hunter with 2 marketed drugs and multiple drugs in clinical development. These include Invanz, an anti-infective approved in 2001, and Mozobil, a stem cell mobilizer approved in 2008. Before joining Expansion, he was the Chief Science Officer and founder of X4 Pharmaceuticals. Prior to that, he was the Senior Vice President of Drug Discovery and Development of Lysosomal Therapeutics, where he invented and developed LTI-291 for GBA Parkinson's disease, currently entering phase 2 development. Earlier, he was the interim head of small molecule discovery at Genzyme and VP of medicinal chemistry, where his group was responsible for identifying Venglustat, which is in numerous late-stage clinical trials. And prior to joining Genzyme, Dr. Skerlj was part of the executive team at AnorMED, a publicly traded company acquired by Genzyme. He started his career with research positions at Merck and Johnson Matthey. Dr. Skerlj is the inventor of 52 patents and is author on over 70 scientific publications. He's a member of the SAB of Vasamune Therapeutics, Cara Therapeutics, and Sperogenics Therapeutics. Dr. Scarole earned his PhD in synthetic organic chemistry at the University of British Columbia and completed postdoctoral fellowships at the University of Oxford and Ohio State University. Also today, we have with us from Expansion, Dr. Matthew Disney. He is a native of Baltimore, Maryland, receiving his BS from the University of Maryland and his PhD from the University of Rochester in physical chemistry. As a graduate student, under the guidance of Professor Douglas H. Turner, he studied RNA catalysis and developed approaches to determine RNA secondary structure in cells by using a combination of experiment and prediction. Matt completed postdoctoral training at the Massachusetts Institute of Technology and Swiss Federal Institute of Technology in Professor Peter H. Seeberger's lab and helped develop the glycan microarray technology. Dr. Disney is currently a professor in the Department of Chemistry at Scripps Research. His laboratory has pioneered the area of small molecule targeting of RNA. A central feature of the lab has been to address fundamental questions of how molecular recognition occurs between RNA folds and small molecules, and to apply this knowledge to address questions of biomedical importance. Applications have included development of sequence-based design of small molecules, on-site drug synthesis in diseased cells, understanding the biology of coding and non-coding RNAs, and working on interfacing RNAs with quality control machinery that has included monomeric degraders and shimmer degrading compounds. The lab's research has garnered various awards, including the Sackler Prize in the Physical Sciences, Barry Cohen Award in Medicinal Chemistry, NIH Director's Pioneer Award, the Tetrahedron Young Investigator Award, the Eli Lilly Award in Biological Chemistry, and the David W. Robertson Award in Medicinal Chemistry. Okay, so it's time to meet Expansion Therapeutics. As a reminder, and thank you very much for all those during the registration process who already submitted questions for the presenters. They have done their best to integrate your questions into their current presentation. And you will also have the opportunity to ask additional questions during the presentation. In order to do that and to get those questions directly to the presenters, please note that you have to use the chat box in GoToWebinar, use the drop-down function to select Send Question to Staff, and that's the only way that the presenters will receive your question. So use the chat box, drop-down menu, Send Question to Staff, and send your question along. Okay, thank you everyone for joining us today. Thank you to Expansion Therapeutics, and gentlemen, take it away.
Good afternoon, everyone. My name is Ronaldo School. I'm the CEO and President of Expansion Therapeutics. I want to thank Tanya and the Myotonia Dystrophy Foundation for the opportunity and the invitation to speak to you today about the research that we're doing, specifically around myotonic dystrophy, although we're also looking at other disease areas. Our mission from the beginning has always been to basically come up with drugs that can treat some of these severe RNA-mediated disease. And in the case of myotonic dystrophy, as many of you are aware, it's an expansion repeat in RNA that we're trying to treat. I'm really happy that Matt Disney will be joining me on this call. He'll be discussing some of the research that we're doing and some of the work that we've done previously in developing drugs to treat myotonic dystrophy. So let me just go to the next slide. So I just want to basically mention a couple of things here. Firstly, myotonic dystrophy we look at as a system-wide disease, which is to say that it treats peripheral symptoms as well as central symptoms. And although, from the outside community is typically viewed as a muscle wasting disease, fatigue and sleepiness are kind of two very common symptoms reported by patients. And because of that, one of the things that we did early on as a company is look to develop a drug that would treat one of the most widely reported symptoms, which is excessive daytime sleep. So what I'm going to do during this presentation is actually talk about some of the top line results we have from a clinical study with a compound called ERX-963 that was dosed intravenously. It's actually a drug that was repurposed. The original drug was called Flumanasal. And this is a drug that was approved to treat benzodiazepine dependency. And these are common drugs such as Valium, Versed, or Xanax. So these are common drugs that most of you are very familiar with. It's used to basically treat overdose in that case, and also to help patients wake up after surgery when benzodiazepine is used as a sedative. The drug actually binds to a receptor in the brain called a GABA receptor. So this isn't a drug that modulates RNA as the other drugs in our pipeline do. It has a novel mechanism of action. And one of the reasons we got into that is that early reports suggested that it was effective in treating idiopathic hypersomnia. And because of those early reports, we thought there was an opportunity here to really treat one of the symptoms that was widely reported to affect patients with myotonic dystrophy. Now, one of the questions that we had early on is, what is a small molecule? So I look at that as an interesting question, just in the sense because I'm a chemist by training. So a small molecule is small, and what I've shown you here on the bottom are two chemical structures. On the far left is a structure of caffeine, which most people I think drink coffee. And you can see, looking at it, even if you're not a chemist, and comparing it to Flumanasal, it actually looks pretty small. One of the attributes of small molecules is that they can be widely distributed across tissue, and in some cases can actually cross into the brain, as both of these drugs do. And we'll take additional questions at the end of the presentation. We've incorporated some additional questions that were raised early on during this presentation and others. And then we have a slide at the end that jumps into a little bit more detail about specific questions that were raised. So the study objectives for this clinical study was to really assess, firstly, the safety and tolerability of a single dose of Flumanasal versus placebo in patients with myotonic dystrophy. The secondary endpoint was to obviously assess whether it had an impact on excessive daytime sleep, and also in terms of working memory in patients, because again, this is another consequence of myotonic dystrophy, is the memory is affected, as well as the daytime sleep. The design of the study is shown here. So just very briefly, we had 9 subjects or 9 patients that were enrolled in the study with a diagnosis of DM1 and excessive sleepiness. They were divided into 2 cohorts. So basically 2 groups of 6 patients per group. One group was treated with 1 milligram of the drug versus placebo over a 5-minute infusion. And the second cohort was treated with 2 milligrams of drug over a period of 15 minutes with infusion of the drug. And the patients were monitored for safety, looking at any adverse events. And the blinded review of cohort 1 safety data prior to initiation of cohort 2 was undertaken to ensure that the drug was safe and well tolerated. Now, also, we took basically PK samples, which is to say that we measured the level of drug after administration to determine what the levels were after 30 minutes or so following each cohort, and trying to basically correlate that with any clinical changes that we saw. So how did we assess efficacy in this study? So we used a standard scale called the Stanford Sleepiness Scale. This is a questionnaire that patients undertook, as well as caregivers. And we also used a patient and clinical global impression of improvement. And again, this is basically based on a questionnaire. There were also additional tests, a psychomotor vigilance test, where a number of parameters were basically assessed to determine whether the drug had an effect relative to placebo. There was also a back working memory test, again, to determine if the patients that were receiving the drug benefited from the drug relative to those that were receiving the placebo. Now, the participants were 18 to 65 years of age. As I said, they were basically confirmed genetically diagnosed with myotonic dystrophy and excessive sleepiness or hypersomnia. And to basically be enrolled for the study, there was a scale, a sleepiness scale that was basically measured greater than 11, or long sleep periods greater than 10 hours per day. Now, some patients were excluded based on general overall health. For instance, if there was significant respiratory or cardiac issues, if there was restless leg syndrome or nocturnal hypoxia, and also depending on the use of medications that the patients were on, or if there was a history of seizures or panic disorder. So really trying to enroll patients that basically had the excessive daytime sleep that wasn't complicated by other health issues. And that's the reason for having the exclusion criteria in there. So if we look at the demographics, so you can see here, just if you take a look at the very far right column, this is looking across both cohorts. So in cohort 1, 7 patients were enrolled. In cohort 2, there were 5. The median age was 51 across both cohorts. It was an equal distribution of males and females. Cohort 1 was skewed towards males, and cohort 1 was skewed towards females, and cohort 2 was skewed towards males. Predominantly, the demographics in terms of ethnicity was Caucasian or European for this particular study. And if you look at the medical history of the patients that were enrolled, so what was the age of diagnosis for most of these patients with myotonic dystrophy? Again, the median age of diagnosis was 38 years of age. The onset of symptoms was around 25 years. And interestingly, the onset of excessive daytime sleepiness or hypersomnia was about 15 years after the onset, so typically occurs later. So the safety is described here. Flumanasal was well tolerated, which is to say that there was no significant adverse events or SAEs. There were some slight or mild adverse events that were reported, typically injection site pain, some headache. 6 of these were unrelated to study drug. And importantly, there were no clinically significant lab values or vital signs that were due to the treatment of Flumanasal in the patients that received the drug. Now, shown on this, chart or bar graph here is, the score response based on the Stanford Sleepiness Scale that I mentioned early on. A negative change from baseline indicates less sleepiness. And you can see by looking at this bar chart that there's very little change across, the dose 1, 2, 3, or 4. So this is just basically looking over time. Is there an impact in the sleepiness scale versus placebo? And again, this is the combined cohorts 1 and 2. So this is both drug doses, 1 milligram and 2 milligrams. And unfortunately, there was no improvement from a sleepiness perspective. Now, looking at this data from another perspective, which is looking at another way to score the data, is using the patient or clinical global impression of improvement. Again, this is basically where the patient fills in a questionnaire to determine how they felt based on drug. And the clinicians or the caregivers also do the same in terms of scoring the patients relative to their sleepiness scale. And so looking in red means that the patients felt better, or better than Flumanasal. Sorry, the placebo was better than Flumanasal. Blue means that Flumanasal was better than placebo, and yellow means that there was no difference. And so if you look at the far left, this is the patient score. You know, some of these are basically, you know, a little worse, which is in red, or a little better. So taken together, and there were 7 patients enrolled in this particular cohort 1, there was really no change here, unfortunately. When the clinicians scored the same patients, you can again see that there was very little change from medium. So no difference at all relative to placebo for 3 of the patients, a little better, a little worse for a couple of other patients. If you look at the data for cohort 2, again, very similar to what we saw in cohort 1. So remember, cohort 2 was a slightly higher dose. It was 2 milligrams versus 1 milligram. And again, you can see, looking at it from either the patient perspective or the clinical perspective, that unfortunately there was very little difference relative to placebo. So I think this is a really a reflection, I think, in terms of how difficult science can be. Expansion made a investment in terms of trying to study Flumanasal in patients with excessive daytime sleep, looking to basically alleviate that symptom. Although the drug was well tolerated, we didn't see any clinical benefit based on 2 criteria, the Stanford Sleepiness Score and the Psychomotor Vigilance Score. And again, this was disappointing. And as a consequence of this, we do not plan to support the further development of extended release Flumanasal to treat excessive daytime sleep in myotonic dystrophy patients. Now, that said, we do have additional research that's currently ongoing, and Matt Disney will describe this to you in more detail in terms of trying to treat basically the fundamental science. So basically what we're trying to do now is really tackle it from another angle and really go back and see if we can actually develop an agent that can modify the disease itself. And so a little bit more challenging, but again, I think it speaks to our dedication to try to find a drug that can treat not only the symptoms, but also the cause of the disease itself. So I'll pass this on to Matt now, and he'll talk to you about the research that we're doing in terms of treating the expansion repeat with a drug to see if we can basically alleviate the system-wide defects that are associated with the disease.
Thanks, Renato. Yeah, so I'm gonna transition here and talk about what Expansion's been working on to design small molecules that target the RNA repeat that causes DM1. And so the reason that we're interested in a small molecule, an orally bioavailable blood-brain penetrant small molecule, is that, as you guys are well aware, is that DM1 is a system-wide disease. So there are defects in the hands and the arms, there are physical health issues, and there are also issues that are associated with the CNS, such as fatigue and sleepiness. And so one medicine that can treat all of these symptoms needs to get in these tissues that are affected, which include the skeletal muscle and also the brain. And so what we're aiming to do is make orally bioavailable medicines that are like routine pills that can treat the whole of the patient, so that can enter each one of these disease-affected tissue with a single agent. And so the way that we're able to do that is we're focused on small molecules that are very small compounds. And because of the nature of their size being very small, that you can swallow them as a pill and they'll enter disease-affected tissues, including the brain. Yep. And so, you know, DM1 is caused by this CUG repeat expansion that's in the 3' UTR of a DMPK message, as you guys are well aware of this. And this repeat was associated with DM1 by studies initially in David Hausman's lab and the students associated with that. The disease is caused because this toxic RNA folds into this structure and it binds to and sequesters proteins that are involved in RNA splicing defects, which is called muscleblind-like 1 protein. And so it's the simple formation of this toxic RNA protein complex that causes DM1, and its main cause is because when muscleblind is bound to these RNA repeats, it doesn't perform its natural function. And so if we can make a small molecule that targets this interface, binds to the RNA, and frees the protein, then that single molecular entity can treat the whole of the disease. And it wouldn't just be the symptoms, it would be the cause of the disease. And it's this, again, it's this simple RNA protein complex that causes disease from the musculoskeletal issues, so contraction and relaxation of your skeletal muscles, to cardiovascular defects, to also issues in CNS. And so Expansion has built a large amount of infrastructure to design and deliver small molecules that target these repeats. What I'm going to show you here is some data with a tool compound. And this tool compound is a medicine that allows us to study how well we can affect this disease-affected pathway in a myotonic dystrophy type 1 mouse. This is the mouse model that was developed by Charles Thornton. And in your muscle, in patients and also this mouse model, what happens is they lost their ability to control contraction and relaxation of some of their skeletal muscles. And the reason that this happens is that in DM1, you lose a key protein that controls that contraction and relaxation. So muscle contraction and relaxation is due to controlling an electrical current. And when you're missing this protein on your muscle fiber, you can't control that current, and therefore you have defects in your ability to control your muscle function. And so what's shown here to the right, again with a tool compound, is if we look at the green staining, that is the expression of that protein that controls contraction and relaxation of the muscle. If you look at the panel that is listed DM1, if you look into this DM1 mouse model, it doesn't express that green protein, as we can see. However, below that, if you look at a wild type mouse, that wild type mouse has broad expression of that protein and has normal muscle function. To the far right is a mouse that has DM1, but it's one in which we delivered our tool compound. And what you can see is that this tool compound allows enhanced expression of this muscle-specific chloride ion channel. And what we've been also able to do is track that increasing this protein expression also enhances the ability of these mice to control their contraction and relaxation, which is shown here. So what this is shown is a video of an untreated DM1 mouse model that has what's called action myotonia. So if you pinch these DM1 mice hind legs, what'll happen is as you pinch it, they have a cramp that they can't relax. So here's a pinch of the mouse's hind leg. And what you can see is it has this cramp in its legs where it can't relax it. It's kept under tension for a long amount of time. And this is an assay that we have in Expansion, and this was discovered by Charles Thornton, who you all know has been one of the key people in developing these mice models and a mechanistic understanding for DM1. If we go to that same DM1 mouse model on the next slide, and we deliver it this medicine that targets the CUG repeats, you can see that this mouse is moving around the cage very quickly. When you go and squeeze its hind legs, it doesn't have the cramp like the mouse without the drug does. In fact, it's able to very quickly alleviate that issue that happens under tension. And when we do this, if we take on day 1, that is before the treatment, all 9 of the mice in this study had that cramping issue. But on day 31 after treatment of this compound, none of them had that issue. And so what this shows is that if one can deliver a compound, a small molecule that targets these RNA repeats, is that you can get alleviation of these defects. And now the reason that I said that this is a tool compound is, as Renato had mentioned, and all of the people in the series are going to undoubtedly mention, is that drug discovery is not easy. It is very hard. And one of the tenets by which all of us have to operate is that we do no harm to the patients. And that do no harm means that all of the compounds that are developed have to go through rigorous testing to have a large window between where they would treat the disease versus where they would have some undesired effect. And that the larger that window is, the more able you are to deliver a medicine that can do no harm and treat the disease. And so although this lead compound was able to alleviate these disease-associated defects, it didn't have a sufficient window by which we could treat the disease and do no harm that allowed us to continue this compound series and being progressed. But one thing that I want to stress here is, and it's one thing in the DM1 community that I've been very impressed by is you guys should understand that the reason that there's a dozen or more people and companies that are invested in this area in making a medicine is because there's a large foundation to be able to deliver that. The foundation of basic science that people like Maury Swanson, Tom Cooper, Laura Ranum, Genevieve Gordon, John Day, Charles Thornton, etc., have laid has allowed for companies, initially Biogen Ionis, to be able to put a medicine into a patient. And Frank Bennett and Stan Cook and Ionis has demonstrated in collaboration with Biogen that biomarkers can be moved if the medicine can be efficiently delivered to muscle. And so we are excited by that path being laid. And the question is, what vehicle do you have to get to put on the path to get to the very end of that journey? And so we're trying to do that. What is different from our company versus the others is that we're making molecules that target the toxic repeat that can treat the whole patient in a pill. And so we're in the early process of discovering new small molecule medicine to treat DM1 that can treat all of these DM1 symptoms, both muscle and CNS. And we expect to be dosing patients in a few years. In addition, and I want to stress this here, is that Expansion has an early discovery program in DM2. So we are addressing both DM1 and DM2. And we have data with early stage small molecules that allow molecules that could eventually be delivered into a pill that can trigger elimination of the DM2 repeat. So it's a small molecule that can be oral that can target these repeats and have an oligonucleotide-like activity that can treat the whole patient. And we're planning to industrialize the medicinal chemistry around this program in 2022. And so, I think that all of these, you don't know that you have a medicine until you have it. But the one thing here that I want to stress is that I think the path to be able to deliver that medicine and the resources to be able to deliver it have been put in this community, not just by Expansion, but all of the other companies that are working in this area. They're very excited by the potential to be able to deliver on the promise that the patients have contributed to by, contributing tissue, contributing to studies here. And so we're very grateful to have the opportunity to be able to do that. And we're very grateful to have Renato, who's really top-notch drug discoverer, to lead our efforts in doing this.
Thanks, Matt. So here's a few questions that were posted early on. So what is Expansion focused on? As Matt said, we're very invested in DM1, and we're starting a program to industrialize the medicinal chemistry on DM2. We're also actually working in other neurodegenerative diseases, and we're heavily invested in trying to develop actually drugs for a lot of diseases where there's very few treatment options available right now. Matt, there's a question posed here about what is our approach to routing the body of the RNA repeats that we have. I think you've spoken to that. I don't know if you want to add any more to that.
Yeah, basically, we have the ability with certain of these toxic repeats to stimulate natural processes. So when your body makes an RNA, it's not like your DNA that you go inherit that, give that to another cell. Your RNA is made to be degraded. And so what we've been able to do is figure out a way to make small molecules that can bind to RNA that cause DM2 and have these natural processes that eliminate RNAs, facilitate the elimination of this DM2 causing repeat. And so we're excited by this because it opens up a lot of opportunities that might make that disease more druggable than could have been appreciated at the beginning.
Thanks, Matt. Unfortunately, the Flumanasal ERX963, we're not working on that moving forward, just given the fact that, relative to placebo, we did not see a benefit. The question regarding our modality, whether it's oral injection, we are looking to develop oral medicines. So these are medicines that are taken as a pill. Our emphasis right now on DM1 is to develop a pill that would treat the system-wide defects, so both the peripheral, which is the muscle, as well as the brain. We're not looking to basically continue working on GABA antagonists. We did that because we thought there was an opportunity to basically treat excessive daytime sleep based on some early results, and we're not looking to basically develop these any further. In terms of clinical trials, in the future, we would basically do, national trials to begin with, and then multinational as the drug moves further into development. So typically phase 2, phase 3. In terms of a genetic test, yes, we would use a genetic test to basically determine that you're eligible to receive the drug for myotonic dystrophy. Will your drugs reverse or stop existing disease progression? I think that's an interesting question. Matt, do you want to take that question?
Yeah, so I don't think you know until you go to do the study. I think there is reason to believe most certainly the compounds would stop disease progression. The question is that when you stop the disease, how much capacity do disease-affected tissues have to repair themselves? And so I think, what we've been able to see early on in the mice is that there's been an ability to have some level of repair. And so I think, it becomes a question that will have to be addressed in the clinical trial. So we're optimistic that most certainly we have significant reason to believe that it'll stop the progression. We'll have to see about if improvement happens and to what extent it can reverse the disease, but we're optimistic.
And then the last question on this page speaks to whether we're looking at multiple treatments. So are you going to basically take the drug as you would a cholesterol lowering agent, for instance, or a drug to basically treat diabetes? Those are basically taken over your lifetime on a pretty regular basis. We would envision the same here with a small molecule, although some small molecules have been developed in certain disease areas where they are administered either monthly or in some cases either bi-yearly. But our first objective would actually be to develop a drug that would very likely be taken once a day to treat the system-wide defects. We've also received some additional questions now. There was one question from a participant asking for clarity. Are there no need for subjects for trials at this time? So yes, to clarify, that is correct. The Flumanasal trial is complete, and we don't anticipate starting another clinical study with our small molecule for a few more years. Another question that came in is, why are some drugs administered via IV and others orally? Is one better than the other? Can you help us understand? I think in the case of Flumanasal, different drugs have what we would say different properties. So, if you take a pill, for instance, you would expect that drug with one dose to work, for several hours and hopefully for 24 hours. For instance, if you take ibuprofen, you typically would take ibuprofen every 4 to 6 hours, because that's the effect that the drug gives you the most benefit. In the case of Flumanasal, it had to be basically administered IV, just because it's not a very good oral drug, in the sense that if it was given as a pill, it would probably get eliminated very rapidly by your body. So it really has to do with the nature of the drug itself. It's not, one's no better than the other. In the case of IV versus oral, it's really a convenience factor. With an oral drug, you can take a pill yourself at home. With IV, you typically need to go into a care facility to basically have IV administration. So really, the biggest difference is convenience. Another question that came in is, will you look at other animal models or just mice? Can you tell us about why mice for DM1, are they important? Matt, do you want to take this one?
Sure, yeah. So the reason, I'll start with the last question first. So the reason that mice models are so important is because those are the ones that have been established by the labs of Charles Thornton, Geneviève Gordon, that have been most developed, where one can see disease defects that models that in a human. And so certainly you want to be able to use those mice models to see, do your drugs get into disease affected tissue? Do they improve disease defects in these mice? Most certainly, we do need to test compounds through development, through other higher level mice like rat and other things, to see how they get into those tissues so that you can better model tissue penetrant in human. Unfortunately, there's no rat or higher level animal model that recapitulates the phenotype of DM1. And so you would really be testing in those other animal models to model the exposure that one would see in human, so that when you start a clinical trial, that you would be able to figure out the dose, and that you would also, again, know that you would, with some level of confidence, that you would be doing no harm in a human based on these other animal models.
Thanks, Matt. We have one last question that's come in, again, just around what is a small molecule, and some examples that folks may be familiar with. I guess, one way to think about a small molecule is, most people have taken aspirin, most people have taken ibuprofen, Tylenol. Again, these are all basically medicines that you take a pill and you swallow. I think that's the best way to think of a small molecule, is something that's taken as a pill that you swallow, and then you either take it one time per day, or you take it multiple times per day. And I think, when you look at some of the other modalities that are basically in development, they have very different technologies in terms of oligonucleotides. For example, those kind of drugs that are administered sub-Q, so they're administered by injection. And I think that's the biggest difference between a small molecule and some of the modalities when people talk about oligonucleotides, or if they talk about gene therapy. These are all basically modalities where an injection has to be given versus taking a pill. So I hope that clarifies it for folks. There is one more question. What's the timeline to get to your new approach to clinical trial with humans? I think that's a great question. Timeline is really driven by so many things in terms of what we do from a research perspective. As Matt showed you some early data on a lead that we had that looked fantastic in terms of the Thornton mouse model where it was used, and unfortunately the therapeutic window was low. The typical time taken from what we would say is when you discover something interesting from a drug perspective to get it into development is about 2 to 3 years. And then once you've identified something that you would basically say, okay, this is something that could be dosed in humans to treat the disease. It takes about another year of regulatory studies, and these are basically very rigorous studies that are mandated by the FDA before we can get into human clinical trials, where you have to manufacture the drug what they call a GMP. So it has to be basically manufactured under conditions that attest to the purity and the efficacy of the drug. So you're looking anywhere from, in our case, given where we are in terms of the DM1 program, I'd say anywhere from 3 years in the earliest to probably 4 years before we can get into treating patients that have the disease. So we do have one additional question that's come in. Do small molecules allow transport of therapeutic agents into cells? The answer to that is yes. So small molecules, by their nature, basically have very good tissue penetration and cell penetration. And again, that's probably an advantage when you think about some of the other modalities. So for instance, just looking at oligonucleotides as an example, these are fantastic drugs, but the challenge there is basically getting wide tissue penetration. So what the small molecule does, just by nature of it being small, is that it can get into wide tissue distribution and can get into cells.
Okay, I'm not seeing any additional questions on our end here either. So it sounds like you answered everyone's questions so far. Thank you so much, both of you. I don't know if there's any last comments that you wanted to make, or if your presentation is complete.
I think our presentation is complete. I would like to thank the patients that enrolled in our study, the study to see whether Flumanasal would have an impact on excessive daytime sleep. And I would like to thank Tanya and the Myotonic Dystrophy Foundation again for the invitation to share what we're doing with the community on myotonic dystrophy and what we're planning to do in the future.
Thank you so much, Renato, and thank you, Matt, also for being here today to talk with us, to share with the myotonic dystrophy community about what's happening with respect to drug development, and for giving us hope, because we know that there's been some ups and downs in drug development for myotonic dystrophy, and it's exciting and it's inspiring to hear that even when one avenue, doesn't work out, that we just turn a corner and we try something new. And that is very hopeful for us, and we are excited that in the next several years we're going to see some big changes. So thank you for helping lead that discovery, and thank you for sharing with us. We will, as a reminder for everyone on online today, we will post this to the Digital Academy. If anyone has additional questions for Expansion Therapeutics, please feel comfortable sending them on to info@myotonic.org, and we will forward them on to Dr. Scarole and Dr. Disney. Hopefully we'll be able to get you some answers more directly that way. Otherwise, we are delighted to have had everyone here, and thank you again. I hope you have a fantastic Friday and a wonderful weekend, and we look forward to seeing you all again soon. Thank you so much.
Thank you.
Take care.
Take care of each other. See ya.
Bye-bye.